so today I want to tell you about start out just by talking about this topic that's quite close to my heart about how we track microbial strains in the human gut microbiome. So this is the agenda for today. We'll talk briefly about what it means to track strains and why we'd want to do that. We'll talk about some of the current methods for how we track strains in the gut microbiome, as well as a method that I and others have developed to use population genetic variations to track strains at very high resolution to infer patterns of microbial transmission. I'll then end by talking very briefly about some of the things that we've learned using these methods, as well as what I see as some of the big upcoming challenges for the field. All right, so first, what does it mean to track strains in the gut microbiome, and why would we want to do that? Well, as I think all of us have probably heard, the human gut microbiome has major impacts on human health in pretty much every aspect of kind of development, health, and disease. And in order to understand the microbiome and its impacts on human health, that requires us to have a really deep understanding of microbial ecology and evolution. In order to understand the structure and function of the gut microbiome, we need to know how microbes interact, as well as how microbes change over both short and long timescales. Now, historically, it's actually been quite difficult to get a view into ecology and evolution in microbial communities. And one reason for this is just because traditional ecology and evolution in plant and animal kind of systems tended to begin with direct observation of the natural world. And so folks like Darwin, could go out there and look at the phenotypic variation among species of finches, and they could infer both the evolutionary relationships between these species, as well as something about their ecological roles, the ways in which they compete with each other for a limited pool of resources. In contrast, when it comes to microbes, we have much less phenotypic variation to go off of. Microbial physiology and morphology provide much less information about either the evolutionary relationships in these communities, let alone any sorts of actual ecological interactions. And so historically, it's been just very difficult to understand what's going on in microbial communities. Now, a lot of this has really changed with the advent of genome sequencing, which I would analogize to kind of like a microscope for the study of ecology and evolution. And so genome sequencing revealed some of the morphological, some kind of variation between microbes that we can use in order to infer some of their evolutionary relationships. And this also gives us the ability to answer even the most fundamental ecological question of which microbes are present in a given community context and how that varies between different settings. And so over the past 50 years, genome sequencing has really revolutionized the study of microbial ecology and evolution. This really started in the 1970s with the development of 16S rRNA sequencing. So for those of you all who aren't really familiar, 16S rRNA is a very highly conserved gene that evolves very slowly across all groups of life. And so by sequencing this gene, we can get a sense of the deep phylogenetic relationships between bacterial clades. As sequencing became more high throughput, we started being able to do 16S sequencing across an entire community. And so this was really powerful because it allowed the field to start assessing community composition using cultivation-free methods, meaning we didn't actually have to successfully grow these microbes in the lab to know who was there and how common they are in these communities. Our capacities have expanded more recently with metagenomic sequencing. Now, instead of targeting just a single gene, we can actually sequence all of the microbial DNA present in a community. And so we can start to ask who's there in a community, how common are they? And we can also start to gauge kind of the functional content of a community using kind of a general bag of genes approach. More recently, a lot of folks have started doing strain-resolved metagenomic sequencing, which is what I'll talk about today. And so strain-resolved sequencing actually just relies on the same shotgun metagenomic sequencing that I showed kind of in the last panel. But the difference here is that we're using new bioinformatic methods to actually try to infer entire bacterial lineages or haplotypes from these short reads. And so the methods for doing that are still under active development, and that's what I'll talk about today. And so altogether, through genome sequencing, we've been getting a broader sense of the kind of breadth of bacterial diversity and the kind of existence of kind of previously undiscovered microbial variation. And we can also zoom into the tips of this kind of tree of bacterial diversity in order to learn more about the fine-grained kind of scales of genetic variation, including the evolution that happens. Okay, so again, this gives us some sense of the depth of microbial diversity within a particular population. All right, so I do want to take a moment to talk about what I mean by sequencing microbial strains. And so our goal here is to investigate diversity within species. In the taxonomic hierarchy, going from domain to species, strains kind of sit below the category of species. And so we're looking at genetic variation that's present kind of within populations. And so what counts as a strain depends really on context. There's not a single answer. It really depends on the application that you're looking for. And that's because genetic diversity within a species exists on a continuum. And so, typically, if you're sequencing near clonal isolates, people usually won't call those different strains, although sometimes you could. Strain instead tends to refer to some level of variation in between individual genomes and kind of distinct species or kind of more deeply diverged clades. But it is a little bit of a fuzzy definition, so I just want to acknowledge that. And I'll define it more precisely for the applications that I'm using. So why should we care about this genetic diversity within species? Well, one is that it gives us the ability to track how strains transmit between hosts and between different environments. And so folks have used these methods to track patterns of transmission between, say, mothers and infants, as well as transmissions that occur during the process of fecal microbiome transplantation. Additionally, strains within a species can actually carry very different gene content, and with that, different metabolic capacities. And so this plot here, I think, is quite cool because you can see that even strains that have very high core genome similarity above the 95% threshold that we typically use to define species, they can still actually have a lot of variation in gene content. And with those variations in gene content usually come differences in metabolic utilization. So these folks phenotyped a lot of strains from different species in the Bacteroides genus. And what they saw was that even within a particular species, like Bacteroides uniformis here, you can see that different strains have different abilities to use different polysaccharides. And so for gauging microbiome function, it's not enough simply to know what species are present. We actually have to understand strain level variation as well. The last reason that I'll mention here is that strains can actually undergo adaptive evolution within hosts. And so this can happen over just kind of the course of host lifetimes. And so really beautiful work from folks here in this room has shown that strains of a species can actually diversify into coexisting lineages, and just a handful of mutations can actually promote this kind of ecological differentiation. And so this really tells us that we need to pay attention to even very fine scale levels of genetic variation within hosts. All right, so to summarize, why should we track strains in the human gut microbiome? Strain level diversity can tell us about the assembly, function, and evolution of the gut microbiome. All right, so how do we track strains in the gut microbiome? So I'll give a brief overview of kind of the current landscape of methods for doing so. There are a lot of methods out there, so if I missed your favorite method, or perhaps even one that you've developed, I assure you it's not intentional. This is just for the sake of time. Feel free to kind of chime in and mention anything I might have missed. And so just to reiterate kind of what we're trying to do with strain-resolved metagenomic sequencing, our goal is to take a bacterial community and just sequence all of the DNA that's present in that community using shotgun sequencing, kind of typical Illumina short reads. And then we want to reconstruct strain haplotypes that can give us a sense of genetic diversity within species. And so ideally full bacterial genomes, but as you'll see, often a little bit less than that. And so the core challenge of inferring strains is from metagenomic data is kind of the meta part of this puzzle. And so people often analogize genome assembly to something like putting together a jigsaw puzzle. And so in that case, metagenomic assembly would be like putting together many very similar jigsaw puzzles at the same time. And these puzzles are also at different frequencies. So you might have kind of 100 times as much of this puzzle as you do of the other one. And so that's actually a tremendously difficult challenge. And I want you to keep this in mind because as I go through very quickly through this landscape of different methods, I want you to pay attention to how these different methods for metagenomic analysis differ and how they handle the meta part of the analysis, as in how reads are actually grouped into different species categories. All right, so how do we track strains in the gut microbiome? Before I get into these metagenomic-based methods, I first want to mention what's still considered kind of the gold standard of strain sequencing, which is just the cultivation and isolation of a particular strain so that you can just take kind of a clonal lineage and sequence it, and so you get kind of often called a pure genome. And so this is still the gold standard because it gives us complete high quality genome. The kind of disadvantage is that cultivation, although it's improved a lot in the human gut microbiome, it's still very limited for a lot of environmental microbiomes. And it also has certain biases, and cultivation is still lower throughput than just pure sequencing-based methods. And so our goal by doing metagenomic sequencing is to reconstruct high quality genomes. And so there are several kind of different families of methods that do so. The first family of methods that I'll mention is kind of marker gene and Kamer-based methods. And so the general kind of logic behind these methods is that we're taking these metagenomic reads and we're severely reducing the diversity that's present within these communities to focus just on a couple kind of well-behaving regions of the genome that are informative about strain level variation. And so StrainFlun and GTPro are some of the kind of commonly used methods in this area. And so the analogy here would be, again, thinking about these genomes as puzzles. We're just focusing in on distinct features of particular species that are different between species, but ideally kind of conserved within species. And so this can kind of help us just kind of focus in on parts of the genome that are really informative about strain level variation. And so typically the first step here is to identify unique species-specific genomic regions. This may be marker genes or, again, just sort of short Kamers. And then we can map reads against this database of marker genes or Kamers to kind of profile composition. And with marker gene methods, we can actually then analyze variation within these marker genes, the reads that are mapped to these marker genes, to gain more insights about strain level variation. And so these methods can be really powerful because they're just pretty fast and efficient at characterizing strain level variation. Probably the drawbacks are that they do require a pretty good reference database, so they work better for some systems than for others, and they are kind of by definition focusing on a very limited portion of the genomes. And so if we think of these marker gene and Kamer methods as kind of reducing the complexity of these systems, then kind of the opposite direction in terms of methodological complexity is full metagenomic assembly. And so this metagenomic assembly actually does involve kind of the goal of reconstructing the complete genome of the constituent bacteria from a metagenomic sample. And so the advantages of this methodology is that these are the only methods. It might be this one that's doing that. Anyway, yeah. So the advantages of this method are that no cultivation is required, and there's also no reference genome database is required, which distinguishes this from other methods. And so this is the primary method used for actual metagenomic discovery, or sorry, new bacterial discovery. And so the first step in these methods is to first assemble or co-assemble metagenomic reads into contigs. And here we use pretty standard graph-based assembly methods that are used for mammalian systems as well. They just rely on looking at overlap between reads to link them into larger segments called contigs. The real kind of tricky step that comes next is that we then want to bin these contigs into, well, we just call them genome bins. And so each of these genome bins is meant to be kind of the equivalent of a bacterial species. And so, oh, wonderful, thank you so much, or our savior. Okay, so each of these genome bins can be reconstructed kind of inferentially by relying on metrics like tetranucleotide frequencies, broader Kamer frequencies, as well as the assumption that contigs from the same genome will tend to have similar read depth, especially across longitudinal samples from the same environment. And so this is the trickiest step of the metagenomic analysis. And so there is a very real possibility of improper binning. It's just very difficult to know whether you've grouped these contigs together correctly, especially because it's often difficult to have a true kind of gold standard or ground truth. And so after we do this assembly, we can use kind of standard methods to predict and annotate genes and then to do other kind of more sophisticated genome analyses. And I want to mention that over the past 5 years, especially, there's been a really great expansion in the use of these metagenomic assembly methods that have really substantially expanded our understanding of diversity, even within the human gut microbiome. And so these methods especially have been applied to samples from populations that have really been under-sequenced and understudied. And so this has really expanded our understanding of gut microbiome variation. All right, so I told you about kind of two methods that are on kind of opposite spectrums of complexity, you know, ranging from full metagenomic assembly to looking at a kind of restricted region of the genome. And so the last kind of family of methods that I'll mention to you is reference genome mapping. And so, this may reflect my particular biases, but I think that reference genome mapping can often be a kind of good balance between the complexity of these two methods. And I'll tell you more about what we can do with these methods. And so very broadly, the idea here is that in order to kind of analyze genetic variation within populations, we're going to rely on the use of reference genomes. And so this is kind of like assembling a jigsaw puzzle when you have the kind of image on the box to guide you. The tricky part, of course, is that the genomes in your sample are not going to be identical to the reference genomes. And so there are just some fun kind of variations thrown in. And so the first step of this analysis is that we map the metagenomic reads against a dereplicated reference genome database. Dereplication just removes genomes that are nearly identical. And so, for example, I do not want a reference genome database with 10,000 E. coli genomes. That would just severely complicate my analyses in unuseful ways. And so here, we just use standard read alignment software like Bowtie2 or BWA, but something to be alert to is the possibility that reads are going to be mismapped between these genomes. When we competitively map between genomes, it's still possible that, especially for closely related genes, that we'll get some reads that by chance are just highly similar and can map equally well to multiple genomes and are randomly assigned. And so we call this kind of both read donation and read stealing. And so this is especially an acute problem when your reference is quite different from the genome within your sample, which you also don't typically know in advance. And so there are kind of additional filters that we can impose in order to kind of limit the influence of this mismapping. Once we've mapped these reads, then we can identify genetic variation relative to the reference genome. And so for those of you who work with human genetics, I think that this kind of family approach is going to feel a lot more familiar. Yeah.
For the reference mapping, do you assemble them reads into contigs before you map them? Or...
That's a great question. So some people do assembly and then they map reads against their own assembly. And so that can be really nice because it removes the possibility of the reference being very diverged. In my case, I'm going to talk about just using kind of standard NCBI reference genomes. And so the reason I like this is just because it's a little bit easier to have the same genomic coordinates across a lot of different samples to facilitate kind of population level. But that is another very powerful set of methods. All right.
Can I also ask something?
Yeah, of course.
What happens with horizontal gene transfer, like in a method like this?
Wonderful question. Horizontal gene transfer just breaks everything I'm going to tell you about. And so that's actually a great kind of preview of my last point, which is that all of these methods currently do very poorly at looking at mobile elements, which are a very large, important portion of the bacterial genome. And so that's a really important caveat to keep in mind. And I'll mention a little bit more how we're trying to deal with that. All right. If there are no more questions, I will continue on. So just to kind of summarize this part of the talk, there are kind of a wide array of methods under active development in order to kind of infer strain level variation from metagenomic sequences. All right, and so for the next part of the talk, I'm going to talk about some methods that I and others have developed to use population genetic variation in order to track strains in the gut microbiome with very high resolution. And so we have a very specific application in mind. A lot of these methods differ because they're used for different purposes. And so our purpose is that we want to infer when past microbial transmission has taken place by identifying strains that are shared between subjects. So when I say that strains are shared, I mean that two people, often two people who live together, have the same species. And when we look at the strains of those species, we may find that these strains are very similar. And so, for example, unrelated subjects typically have strains that are 99 to 99.9% similar in the core genome. But when we start to look at people who have lived together, we occasionally find these cases in which we have these near identical microbial strains with anywhere from 0 to just kind of like dozens of genetic differences. And so this really high similarity of strains between different people suggests that there's been some sort of prior strain transmission either between these subjects or from a common environmental source. And so the goal of the methods I'll talk about is to determine when strains are at this kind of near identical level of similarity such that we can infer that a transmission occurs. Okay, so there are already some folks who are applying this. So for example, this large-scale study came out earlier this year. And so they showed that there was quite a bit of strain sharing within households, particularly between parents and their offspring, but also between adult partners. And so I'll talk a lot more about this in the seminar part of the talk. All right, so in order to identify shared strains, the first step is that we want to infer for two different people the dominant strain that they have of each species. And so the methods that I'll talk about today are kind of based on they use the MIDAS suite of software tools from Katie Pollard's lab. And this kind of methodology has been refined by Nandita Garud and Ben Good, although we've extended these methods as well. And so again, as I mentioned, the first step is to map metagenomic reads against a dereplicated reference genome database. And then we want to identify genetic variation relative to the reference genome. So all of that seems very straightforward, or it might seem straightforward, but one of the challenges that we run into is that subjects can carry multiple genetically distinct strains of a single species. And so, for example, if we look at the Bacteroides vulgatus population within a subject, we might find, say, that there are 3 different strains, which are not fully distinguishable here, but I promise you they're different colors. And so, you know, these 3 strains can make up different proportions of this overall population. What that means is that if we take all of the sequencing reads from the sample and we map them against the B. vulgatus genome, then we would expect to see lots of polymorphisms. And our challenge lies in determining which of this large suite of polymorphisms are actually derived from the same haplotype. And so I'm going to use haplotype and strain kind of interchangeably in this part of the talk. And so there is an analogy here to phasing human haplotypes. So of course, when we sequence humans, we also expect to be sequencing 2 distinct chromosomes, sometimes more if you're sequencing cancer samples. But I would argue that there's some additional challenges we face in the microbiome space. In particular, our haplotypes, our strains, we expect them to be present at equal frequencies in kind of typical human samples because we have 2 chromosomes that are, you know, present in 1 copy each. But we can't make that assumption with strains. As I've kind of alluded to here, strains can be at whatever frequency they like to be. And so we can't make that assumption that things will be at equal frequencies. The second thing is that human haplotype blocks are already pretty well established through extensive sequencing of the human population. Bacterial haplotype blocks are not, and due to the kind of extensive nature of bacterial recombination, it's also possible that this is just not something that we can expect to know. And so I just want to mention that kind of a lot of this kind of looking at strain phasing and things like that, this is prior work by Nandita, who now has her own lab at UCLA, and Ben Good, who I'm collaborating with at Stanford. Okay, so one of the insights that Ben and Nandita had is that if you look at the site frequency spectrum for a bacterial population within a sample, you can learn something about the number of distinct strains within a subject. So for those of you who aren't familiar with site frequency spectra, the idea is just that we take the kind of, oops, that's going too fast. We just take the frequency of different alleles. In this case, we're only using the major allele, so the more common allele of 2 at a site. And then we're counting the number of sites that have alleles at that particular frequency. And so for this particular example, we would expect to find polymorphisms at all of the frequencies that I've listed there. And the reason you can get kind of these combinations of frequencies like 65% is because you can have polymorphisms that are shared between multiple strains. And so basically, when you see a kind of bumpy site frequency spectrum like this, that tends to indicate that there is a lot of polymorphism and probably a lot of different strains. Looking across bacterial populations, then we can look at the different site frequency spectra and identify what Ben and Nandita called quasi-phasable populations. And so quasi-phasable is a nice fancy word. I like to refer to it as easy to look at populations because they consist primarily of 1 dominant strain. And so we are not solving the strain phasing problem. We are just finding samples where we don't have to solve it. And fortunately, that's actually a lot of samples, it turns out. And so this example that I showed you earlier would be not quasi-phasable. We can't actually assign these different polymorphisms to different haplotypes. This sample here has a kind of peak of polymorphisms present at 50% frequency. We also actually can't can't phase those two strains. Even though there's only two strains now, we can't figure out which of these polymorphisms goes with which strain because they're at equal frequencies. So this is not quasi-phasable. Now, the thing is, if one strain is at a much higher frequency than another one, then it does become quasi-phasable, which is great. And so, for example, if one strain is much more common than another, then all of the genetic variants that are associated with this red strain are the ones that are going to be present. Sorry, all of the sites that have variation involving the red strain are going to have a little bit lower frequency, but by taking the major allele, we can still figure out which of the polymorphisms belongs to the dominant strain. And so this is great because we can then infer the genome of this dominant haplotype. And then, of course, there are always, there's actually a surprising number of these kind of trivially quasi-phasable samples where you basically see very little variation aside from what we think might be sequencing error or just recent polymorphisms. And so those are easy to phase. Okay, and so a kind of more formal definition of quasi-phasable populations is populations in which the major allele has greater than 80% frequency at more than 99.9% of the genome. And so again, the kind of intuition there is that most sites have very limited polymorphism, indicating that there's a limited number of strains. Okay, so we have found our quasi-phasable populations, and we've inferred the haplotype of the dominant strain. So now one thing that we can do is to count the number of genetic differences between the dominant strains, and that can tell us how diverged the dominant strains are. And so in this cartoon example, there would be 4 differences, between these 2 strains. And so next, what we need to do is to actually make sense of this number of genetic differences. Is 4 a lot or a little? It's hard to say. So to infer whether transmission has occurred, what I've been doing in the study that we recently released is to compare the number of genetic differences in populations from the same household and to compare that to populations from different households. And so for an example, I'm showing you the distribution of the number of genetic differences between the major haplotypes for pairs of samples that are from the same subject over time, the same subject over time, subjects from entirely different households or different subjects from the same household. And so you can see that strains from different households are typically quite diverged, although there are some cases in which they are kind of more highly similar. When we look at strains from the same subject, they're almost always very, very similar to one another. There's very few differences. And when we look at populations from different subjects in the same household, we see this very nice bimodal distribution. And so this is consistent with the expectation that for some people, they actually have had a strain transmission and then potentially subsequently some within host evolution that causes some limited amount of divergence. And then you also have people who, although they both carry the same species, just acquire different sources. And so this is one method for inferring when transmission has previously occurred within these populations. Okay, but there is another scenario that we want to consider, which is what happens if a shared strain is actually present at low frequencies in one subject. So, for example, we can consider the case when this strain with the red and blue polymorphisms is present just at very low frequency within this other person's population. And so in this case, what we would want to do is kind of specifically to look for the SNPs that are coming from this known haplotype of interest, this kind of other strain that we've already inferred the genetic composition of. And so what we can do is to, we call it fishing. We should come up with a better name, but we call it strain fishing for SNPs from a known haplotype of interest. And so if you want to read more about this, this is kind of buried in the methods section of this paper. This is not kind of a named method or an algorithm or software yet. It's just something we are doing to analyze our data. But if you would like to apply it to your data, please feel free to get in touch with me or Ben. I'd be happy to help you with that. Okay, and so the first step in trying to look for these SNPs. Oh, sorry, go ahead, Tim.
I know the folks who developed InStrain also kind of look for shared polymorphism. Is the fishing sort of fundamentally different than sort of what's going on in InStrain?
Yeah, that's a great question. So the method that I just talked about with the kind of dominant haplotypes is kind of looking for fixed differences, which is the method behind InStrain. And so,
no. Sorry, InStrain actually looks for shared minor alleles. So it says between 2 samples what are all the alleles that are
okay.
shared.
Yeah,
so
maybe you want to. That's my mistake there. Yeah, so we haven't directly directly implemented instrain. Kind of my understanding of that methods was that they were looking for the number of fixed differences, right? And so that's a little bit different than the kind of genetic differences between the major haplotypes. And so, they are also able to detect some of these minor genetic variants. Actually, what we found and kind of what motivated us to develop this set of methodologies is that the in-strain method actually works very poorly when you have a lot of multiple strains present within a subject, just because in-strain is looking for the absence of fixed differences, and you can actually get an absence of fixed differences just because you don't have fixed sites within the sample because there's a lot of strains. Okay, yeah, thanks for asking that. Hopefully that cleared things up a little bit. Okay, and so in our methodology, we aim to identify private SNPs within a haplotype that are actually not detected within the Human Microbiome Project. And so the idea here is that although there are a lot of SNPs that are present in a haplotype of interest, not all of them are actually going to really tell us a lot distinctively about whether the strain of interest is present in the sample that we're looking for. And so again, for a particular haplotype of interest, we can compare the SNPs that are present in that haplotype of interest to the dominant strains of populations from the Human Microbiome Project. And so the idea here is that this is a reference cohort. And so we can identify sites in that haplotype that are what we call public SNPs. So these are sites of just generally segregating variation. And so these are kind of sites where many strains carry polymorphisms. We can also identify sites that are private SNPs. And so these are sites of unique variation that are really characteristic of our particular haplotype of interest. And so I like to think about these sites as kind of like a unique fingerprint for that strain. It represents very recent genetic variation that we're unlikely to find elsewhere within kind of just other circulating bacterial lineages. And so we're going to be focusing on these private SNPs in order to determine if a strain is present in a population. And so once we've identified these private SNP sites, we can then kind of fish for these alleles and see if they're present at frequencies that are above the sequencing error rate. And so, again, we can use this kind of comparison for samples within the same household or different households that I told you about. And so we find, as you would expect, that samples from the same subject tend to share basically all of their private SNPs over time. Or samples from different households have only a very limited amount of strain sharing. And once again, you can see that these samples from different subjects in the same household have this bimodal distribution, indicative that while some people share strains, others have acquired the strains from different sources. Yeah.
Can you clarify, Catherine, like how long do people have to be in the same house to be a same household?
Oh, we consider in this kind of operational definition, we're just saying anyone who is currently kind of cohabiting. So maybe not like people who are sharing a room for a night, but like people who do kind of reside in the same location. But you'll see a wide spectrum of variation depending on how long you've lived together and other factors.
You mentioned that you looked at the variation over time. So what's the time component in this case?
Oh, that's a great question. So here we're actually not looking at variation over time. We do have samples from the same subject taken over different time periods. And that time period could range from, just a few months to actually up to 2 years. But that time component is not kind of something that we're taking into account in order to determine if strains are trend.
Following that question.
So following that question, like the bimodal distribution you saw here, like can you look at the time course data to track like how dynamic that would be, or is this like this distribution, how stable the shared private SNPs are in the same subject?
That's a great question. And so as this kind of same subject distribution alludes to, people do tend to carry the same strains over time. And so these private SNPs, like if we were to look at samples collected from like a household pair, like this year and next year, it tends to be the case that they are still they remain in this part of the distribution or they remain in this part of the distribution. In terms of strain swaps, we do see those as well. I'll kind of allude to that briefly, but those are relatively rare events. But that is exactly what I'll be talking about later.
Thanks. One other question.
what happens, or how do you consider, or do you consider duplicate gene events? So we do have a lot of duplicate genes, and if I now have one bacterium genome and it has 3 genes and one has a SNP, these are the normal ones, then based on this method, it would appear as a low frequency strain or something like that, right? But it's still the same.
Yeah, that's a great question. So we typically screen out regions of the genome that have kind of abnormally high or low coverage. And so we are explicitly kind of trying to look at slightly better behaving regions of the genome. But that is kind of a, that does mean that we lose some information, as I'm sure you can imagine. All right, so those great questions. If there's nothing else, I will go ahead and barrel forward. And so with this method of strain fishing, as well as kind of this kind of method that's based on in-strain, we can detect shared strains with very high sensitivity and specificity. And so we're kind of utilizing more of the genome to do our analyses on a lot of these marker gene methods. At the same time, we're not going to full metagenomic assembly, where we would have to deal with a lot of just kind of assembly issues and gaps in the genome. And so overall, what this allows us to do is to characterize the proportion of strains that are shared between people who live in the same household, which is this kind of red distribution here, versus people who live in different households. And so I'll talk a lot more in the seminar part of this talk about what we're doing with that information. I want to mention that this methodology that we've developed can actually also allow us to analyze the genetic differences between shared strains that accumulate after transmission. So we can see 2 people share a strain, it's nearly identical, but there's 12 genetic differences. We have actually not actively looked into that in our data, but that's something that we can do with these reference genome-based methods. All right, so altogether, I've told you about kind of this methodology that we've been developing to use population genetic variation to track strains in the gut microbiome. And so again, we map reads against a reference genome database, identify quasi-phasable populations with limited genetic variation, where we can infer the identity of the dominant strain. We then identify private SNPs within that dominant strain that are not detected in the Human Microbiome Project. And so for these private SNPs, we then look for them in both cohabiting and non-cohabiting subjects, and we can set a threshold that allows us to determine when we think strains were transmitted. All right, so with the couple of minutes I have left, I want to briefly comment on things that we've learned from tracking strains in the gut microbiome. And so one thing that we've learned is just how many strains per species tend to be present in the gut microbiome. This may seem like a little bit of an esoteric question, but I think it hides a lot of interesting ecology. So again, some really nice studies of B. fragilis have shown that Bacteroides fragilis in particular is a species where you almost always have, you almost never have more than one strain. You either have 0 or 1. And that actually leads to some interesting questions about why it is that there's exactly 1. And there's some evidence that there are active methods of competitive exclusion that prevent co-colonization. Looking across different species within the human microbiome, which are shown on the x-axis, when people have counted the number of strains, which is shown on the y-axis, you tend to find that most species have pretty few strains. It's often 1, sometimes 2, rarely 3. This actually is something that I kind of alluded to with the fact that many populations in the gut microbiome are quasi-phasable. We couldn't just kind of ignore all of these multi-colonized events if, say, everyone typically had 12 or 30 strains of a particular species. And so why is there this kind of phenomenon of oligodominance where you tend to have a few strains of each species rather than, again, like 30 or 100 or something like that? Well, I think this points to some interesting questions about how much strains overlap in resources. You can reasonably assume that strains of a species have a lot of resource overlap, and so it's very difficult to sustain the maintenance of 2 strains of a species very stably. And again, there also may be kind of active mechanisms of competitive exclusion as well. Another kind of question that we're starting to learn about is how do strains respond to environmental perturbations? So here I'll show you a little bit of this nice study from Mike Snyder's lab of one individual over the course of Lyme disease and subsequent antibiotic treatment, which is shown in red. And so in this data set, what the authors found was that there were some species that could remain quite stable over time. This is the proportion of the population that consists of Eubacterium elegans. But under the hood, if you look at strain level variation, you can actually see major shifts. And so here, this is kind of a complicated plot, but each of those lines represents a polymorphism. Just pay attention to this black line here, which shows kind of the frequency of one of the strains. And so you can see that it hangs out at about 10% frequency before antibiotics. After antibiotics, which are here, it shoots up to high frequency, but then kind of declines again. And so this points to some interesting phenomena. Basically, you can have two different strains of a species that actually respond very differently to a perturbation. And this is the case even if we don't see any changes in the species level abundances. So again, I think this highlights the importance of looking at strain variation. And I'll tell you more in a moment, if you stick around, about what we've learned from using this method to just track strains, strain transmission within. All right, so what's next? I will just comment on this very briefly. I think one of the major unsolved challenges for strain-resolved metagenomics is the fact that we have a very difficult time linking mobile elements to their host genomes. So all of the main kind of metagenomic methods fail at this. These kind of marker gene and k-mer methods definitionally do not look at mobile elements because they are not uniquely indicative of strains and species. Reference genome methods fail at this because you don't necessarily expect a mobile element to be present in the reference genome, and the issues with read mismapping are just incredibly high for mobile elements. And then for metagenomic assembly, this is also a problem because these mobile elements just tend to break your assemblies. So you will have a lot of breakpoints and config around when those mobile elements take place because you can't definitively resolve the kind of genome assembly graph. And so mobile elements, as I kind of alluded to, we expect to be quite important for bacterial function and evolution. So what can we do to tackle this challenge? Well, there are some novel genomic methods out there that I think can provide us some methods to resolve this question. One is proximity ligation, which allows us to physically link pieces of DNA that are present in the same cell. We get these chimeric reads formed from different kind of pieces of DNA that get fused together. And then by sequencing these chimeras, we can determine which genomes were actually present previously, or which fragments of DNA were present in the same cell. And this is, I believe, the only method that I am aware of that can actually link plasmids to their host genomes, because there's no other form of kind of physical linkage. Another method that I think is becoming more popular is nanopore sequencing of kind of direct genomes from gut microbiome samples. And so here, long nanopore reads can actually span entire mobile elements, and they can contribute to these really complete genome assemblies. And so this can be really, really powerful. Especially in scaffolding genomes. The main drawback is that right now, nanopore is still more limited in throughput, and given that the relative abundances of species in the microbiome are very skewed, it's still hard to use this for rare species that you haven't isolated. But that problem may become less of an issue as the kind of throughput of nanopore increases as well. All right, so overall, just to review what I've told you about today, we've talked about how tracking strains can inform us about microbiome assembly, function, and evolution. We've talked about kind of the suite of methods available for strain-resolved metagenomics, and a method that I and others have been developing to use population genetic variation to track strains with very high resolution. What we've learned is that there tend to be relatively few strains of a particular species present in the gut microbiome, and also that those strains can behave differently in response to the same perturbation. And then finally, I think some of the open questions involve kind of linking mobile elements to this gene. Welcome back. Thank you all for sticking around. I'm very much looking forward to telling you about the more kind of biological aspect of what I've been doing here on studying the dynamics of colonization and transmission in the gut microbiome. And so I work very closely with 4 different labs as a postdoc, and the work that I'll talk about today is primarily coming out of close collaboration with Ben Good's lab, and then I'm also, of course, in David Relman. As I think all of us know, each of us is home to trillions of microbes that together make up the human microbiome. And so these microbes, especially the ones that reside in the human gut, play key roles in human health and disease. So much so that the role of the gut microbiome has been compared to that of other human organs. Now, of course, there are very key differences here. Our organs are made up of our body's own cells. They form structures through developmental processes that have been honed through millions of years of evolution. And in contrast, our gut microbiome is kind of spontaneously assembled. We're all born microbe-free or very close to it, and then we acquire our gut microbes from the people and places around us. And so I think it's really incredible to think about that. That's incredibly important community within us is so strongly influenced by our environment. And so new microbes shape the gut microbiome over the course of our lives. And so transmission is especially important in the initial assembly of the gut microbiome in early infancy. There are microbes that come from the caregivers during and after birth, often kind of play a major role in shaping the gut microbiome in early infancy. The microbiome undergoes a pretty repeatable shift with the introduction of solid foods. And then that starts the kind of gradual transition to a more adult-like community. Diversity and complexity of the gut microbiome increase over the next several years of life until the gut microbiome reaches a more adult-like composition. And so what do I mean by an adult-like composition? Well, I think one of the best views of that comes from the study now almost 10 years ago from Eric Alm's lab. And so in this study, these two adults sampled their gut microbiomes over the course of about a year. They took nearly daily samples. And so you can see a couple of key features of adult gut microbiomes. These gut microbiomes, first of all, are diverse, meaning they harbor many different residents. They're also stable, meaning that the same residents tend to be present over time, notwithstanding certain fluctuations. And there is also a strongly individual component to the gut microbiome that arises kind of through this combination of diversity and stability. And so all this paints a picture of the adult gut microbiome as a very mature, stable, established community. Now, within this mature community context, there are still opportunities for change in the acquisition of new microbes. And a lot of that evidence comes from studies of human households. So as I already talked about earlier, people who live in the same household sometimes carry genetically near identical gut microbial strains. So this can be kind of over something like 99.99% genetic similarity, near identical strains. And we take this as evidence that there's been some sort of recent transmission either between cohabiting subjects or from a common environmental source. And so recent large-scale studies have characterized the extent of this strain sharing in hundreds of households across the globe. And so they've seen, for instance, that among parents and their offspring, as well as between siblings, you tend to see a lot more of this strain sharing than you do between subjects coming from who live in different households. And so this is all in line with what I told you, that these environmental influences play a major role in the initial assembly of the gut microbiome. I think what's a lot less appreciated, though, is that when these authors looked at cohabiting partners, so adults who live together but are not genetically related and did not grow up together, there's also still a substantial amount of strain sharing. It's actually on the order of the influence of kind of parents on their children's gut microbiomes. And altogether, I think this really highlights the fact that we're continuing to acquire new microbes over the course of our lives. Now, although we can see evidence of these past colonization and transmission events, one major gap in the field is that so far, very few studies have actually successfully captured colonization in action. And I think this is really important because the fact that we don't know this means that we still know relatively little about the ecological context in which these colonization events tend to occur. And so today I'll talk about my attempt over the past 3 years to do that, and what we've learned from this study about how new microbes can colonize established gut microbiomes. And I want to make the case to you that by studying colonization dynamics, so actually tracking the frequencies of colonizing strains over time, we can learn something about the ecological barriers that make it difficult for new microbes to colonize established. And so before I dive into the actual study, I want to take a moment, or actually several moments, to talk more about this context, about this concept of ecological barriers. So what are possible ecological challenges that species might face in colonizing a new ecosystem? Well, I think actually often in thinking about ecology, it helps to think about macroscopic systems. So let's think, for instance, of two islands within an archipelago that have distinct but related ecosystems. We can follow the journey of a new species, like a rabbit, as it disperses from its initial home community and tries to colonize a new community. And so the first step here is just dispersal. The rabbit has to physically make it from its first community to this new environment. And that process of dispersal involves some amount of environmental selection. In particular, in this case, we see this exposure to salt water in hospitable environments. And so all of the very few individuals are actually going to survive these kind of environmental barriers to even arrive within this new environment. After they arrive, their challenges are still not over. So new arrivals are going to face a lot of challenges in establishing viable populations. And I would argue that these are especially acute for newly arrived individuals when the kind of overall population of the species is low. And so for these new arrivals, ecological drift is very high. There's a just very large chance that they'll die out before leaving offspring. And they're also very subject to priority effects. These are mechanisms that favor the resident microbes, and they often have to do with something like, the resident species. And priority effects often come from something like strength in numbers. And so new arrivals to a new ecosystem are going to have more trouble finding mates in order to reproduce. They also can't take advantage of the social structures that tend to increase their chances of survival. And so overall, it is a really strong challenge in order to establish kind of viable populations. Now, even if a new species manages to overcome all of these barriers and establish viable populations, it still faces questions of its long-term survival within this new ecosystem. Overall, its long-term survival is probably going to be shaped a lot by its ability to compete for resources against other resident species and its ability to escape predation. And so overall, this concept of niche availability can really shape whether or not a new species persists long-term in these new environments. All right, so these are all kind of concepts from invasion biology and macroscopic ecosystems, but we can easily import them into a microbial context as well. So when we think about microbes that are colonizing a new host, we have similar ecological barriers. Microbes first have to disperse from one host to another, and in doing so, they experience kind of passage through harsh environments. They encounter oxygen, and they also have to survive the passage through the harsh environment of the stomach. Even after arriving in the gut, these species still face a lot of challenges in establishing viable populations. And so they're subject to ecological drift, and they have trouble forming social structures like biofilms that may increase their chance of survival. And then again, long-term, the survival of these species is going to depend a lot on niche availability and the ability of new species to compete for resources against existing residents. And so these are kind of the direct analogies of those macroscopic ecological barriers, but I want to point out that host-associated communities also have their own particular challenges. In particular, the host is actually constantly detecting and responding to microbes, and so that layer of host selection may also influence which microbes are able to colonize. Although we don't really have data on this aspect, so I won't talk much about that today. Okay, so although these are all potential ecological barriers to colonization, we want to understand which of these ecological barriers are actually most important in the gut microbiome. And much of the field has focused so far on the influence of niche availability. And I would argue that either explicitly or implicitly, many people kind of view niche availability as the main determinant of colonization. And so that comes from studies, classical studies like this one of colonization resistance. And so these authors tried inoculating healthy mice with salmonella. They found that a very large dose of salmonella was needed to establish viable infections. But if they gave the mice an antibiotic that knocked down the resident community, then suddenly it became much, much, much easier for these salmonella to colonize. And so we can interpret these results under this framework of niche availability. It seems like in conditions of low niche availability, it's difficult for new microbes to colonize, but then when more niches are available, like after a perturbation, it becomes a lot easier. And so this framework has kind of been supported by more recent studies as well of probiotic supplementation. And so in this study, authors gave a very large dose of probiotic bifidobacterium to healthy human subjects, and they wanted to identify microbiome differences between the subjects where this bifidobacterium persisted versus the subjects in which it did not. And so the differences that they found, you could link to niche availability. In particular, in the people where bifido successfully colonized, they tended to have less bifido to start off with, and fewer of the genes that utilize the carbohydrates that bifido tends to consume. And then we have the inverse, of course, for the individuals where bifido did not persist. They seem to already have that niche. And so all of this kind of paints a picture that I think few of us will disagree with, in which niche availability is very necessary for colonization. But there are still open questions, to my mind, about whether niche availability is actually sufficient for colonization in human settings. So if you focus just on the studies that I told you about, you would argue that yes, niche availability is probably just all there is. It's really sufficient for colonization. And in that case, what you would expect is that when niches become available, like after a perturbation, colonization should be very fast, and the amount of historical contingency in colonization should be low, providing that you're sampling from the same kind of environmental sources. And so this is kind of like a strong ecological selection regime. But you might also have caught on to the fact that the studies that I highlighted for you are all examples of controlled microbial introduction. And so in those cases, the authors, for understandable reasons, have directly circumvented a lot of the processes of dispersal and things like that. And so you can also argue for an alternative viewpoint that no niche availability is not all there is. And in natural human settings, it might be that actually dispersal, environmental selection, priority effects, all of these may play major roles in limiting colonization. And in this case, you would expect that even if niches are available in the gut microbiome, colonization may still be quite slow, and the historical contingency of colonization might be quite high, simply because you don't have the same microbes reaching the gut, or resident microbes may just have really strong advantages over any new colonizers. And so in my work, we sought to distinguish between these two different views of colonization. And to do that, we set up a study to evaluate the dynamics of colonization after a niche clearing perturbation. And so several years ago, I worked with this really incredible undergrad, Jordan Goldman, who's actually now applying to a graduate program. So keep an eye out for him. And so we recruited a cohort of 22 households of cohabiting adults. And as Ora alluded to, we did this right at the beginning of the COVID pandemic. And so these adults sampled their gut microbiomes. They collected weekly samples over the course of about 2 months, as well as more frequent daily samples in the middle of that 2-month span. And the reason for that more frequent daily sampling was that we gave one person in each household a short course of the antibiotic ciprofloxacin. And so this was purely as a microbiome perturbation, not for any clinical indication. This is a broad spectrum antibiotic, and we gave them kind of the shortest commonly used clinical dose. And so these subjects also continued to collect follow-up samples for up to 2 years after this initial antibiotic perturbation, which allowed us to evaluate some of the longer-term dynamics of colonization after this perturbation. And so just to make this really clear, what we wanted to do was to compare the dynamics of colonization under conditions of low niche availability, so before antibiotics and in the controlled subjects, as well as under conditions of what we infer to be high niche availability immediately after an antibiotic perturbation. All right, and so we use strain-resolved metagenomic sequencing to distinguish resident strains from new colonizers, and I'm very happy that I don't have to talk more about that right now because I just talked about it for an hour. All right, okay, so what are the dynamics of colonization? We kind of break that down into a couple different questions. First, who colonizes after this antibiotic perturbation? We expect to see some losses of species after antibiotics. Do we tend to see the same new species colonizing? And second, how quickly do these new species colonize? As I mentioned, that can tell us about the relative influence of some of these ecological barriers to colonization. And then finally, our household setting gives us this kind of unique opportunity to look at the repeatability of colonization dynamics and whether strains that were previously shared between subjects actually wind up recolonizing after antibiotics. All right, so with that, let's dive into the data. So here I'm showing you, just for your reference, the gut microbiome of one of our control subjects over this 2-month sampling period. And so in this plot, the ribbons show the abundance of different bacterial species within the community, and the colors represent distinct bacterial families. And so what you can see is that the species present at the beginning of the study tend to be present at the end as well, although there are more fluctuations. And I just want to mention that this kind of spikiness in the middle is just a visual artifact. We have more frequent samples collected then. Okay, so how much new colonization happens over the span of time? Well, it turns out that it's actually quite limited. And so here's a plot that we use to summarize the total amount of the gut microbiome that's comprised of new colonizers in each of our control subjects. It's actually a very difficult plot to show because you can't really see anything there, but that's part of the point. So each of our 26 control subjects has a line that is in this cluster right at the bottom, indicating that we detect no new colonizers or just very, very low abundance new colonizers over this 2-month span of time. And you might think, okay, maybe 2 months is not that long of a span of time in the scheme of things. But if we look out over our follow-up samples, which were collected over 1 to 2 years, we actually continue to see very limited new colonization. And so this supports this view that the gut microbiome of adults tends to be quite stable and to show strong colonization resistance in the absence of perturbation. Yes.
That's very cool. In this study, did you like make like have a fixed diet or
Not at
anything
all.
like that?
These were,
No.
yeah. We didn't ask these subjects to do anything different other than collect their poop and also take an antibiotic if they were in that group. So these are humans in the wild for better or for worse. Yeah.
Sorry, just to follow up on that, were they in the wild or were they, you said this was started during the pandemic, so.
Well, yeah, they were doing whatever they were doing, which was probably quarantining.
Yes, so was that through the whole 2-year period, or was that more in the, sorry, I'm sorry, if you can just put a years on those for me.
Yeah. So we had one wave of subjects starting in March of 2020 and the second wave starting in about August, September. And so by the end of the 2-year period, they were done kind of back at work out and about. Yeah, it's a very good question of how that might impact our results. But I'll argue, we can talk more about this at the end, but I actually don't think it has too much.
Can I just ask maybe a question to the first talk? Like in the process of tracking strains, so how many species do you actually on average track strains from per subject? And so that enables you to do this analysis?
Yeah, we sequenced about 10 gigabase pairs per sample for 4 samples per subject. And then we also did lower sequencing for a lot of additional samples in the interim. And so that allows us to track strains with, so we get, above 5x genome coverage for most species above about 1% relative abundance, which winds up being usually, I want to say about 24 species per subject. Certainly we're still missing a lot, but this was already quite a lot. All right, great. So overall, the main takeaway of this slide, as I mentioned, is that as we expect, healthy adults show stable microbiomes and strong colonization resistance. So what happens when we give people an antibiotic? Well, we see this dramatic shift in microbiome composition, which you can see with the antibiotic here. And so about 3/4 of the subjects in our study had what we call a transient response, in which the microbiome shifted during antibiotics, but then returned to something closely resembling its prior state after the antibiotics ended. And so we can break down this transient response by kind of looking at the kind of species behaviors that give rise to it. And so within these subjects, we saw that many species were disrupted by antibiotics. So I'm going to show a lot of these species abundance plots. And so here, this is just the proportion of the community that the species makes up. And you can see that for both of these species, their abundances decline drastically to undetectable levels after antibiotics start, which is the period in gold. After the end of antibiotics, though, these 2 species show different behaviors. And so the first species appears to go locally extinct. We just don't detect it again. But then other species actually do recover to something close to its initial relative abundance. And I want to emphasize that because we're doing strain resolved sequencing here, we actually know that the strains that were here after antibiotics are the same as strains that we detected pre-antibiotics. And so this is really important. It's not that these people are getting kind of new strains from the environment driving this recovery. It's actually their own strains driving the recovery. Overall, among disrupted species, we saw that about 30% of them were lost within these subjects with transient responses, and most of them recovered. And the disrupted species represented anywhere from 1/3 to 2/3 of the species that we detected. And so this indicates that there are, yes, go ahead.
Sorry, following this plot, is there any filter applied to this plot? So because I didn't see Enterobacteriaceae or Proteobacteria inside after antibiotic
Great question. So we do detect enterobacteria. Proteobacteria are a larger group, right? And so they're not going to show up in this family level characterization. I did subsample the number of names just for this line, but they are around. We're not throwing out this family. But we don't see them at high abundance within these individuals, if that's your question. Yeah. Great. Okay, so we do see some amount of disruption and species losses, and so we expect that to mean that there's some increase in niche availability. And so then we might wonder, do we detect additional colonization after these niches become available? Well, again, here are our control subjects where we see very little additional colonization. Interestingly, when we looked at these subjects with transient responses, we continued to see very little colonization over the month before and after antibiotics. They actually continue to look near identical. We do see this expansion of species here, but they're actually ones that we can previously detect. If we look over longer timescales as well, we continue to see that colonization is limited even after this antibiotic perturbation. We do have two subjects who each acquire one high frequency strain or species, but overall, this population actually looks very similar to the controls who didn't take antibiotics at all. And so this is really interesting because it suggests to us that in about 3/4 of the subjects who take antibiotics, we see this extensive recovery of resident strains. We see some species losses, but these species losses don't seem to be accompanied by additional colonization. And so instead, it seems like this rapid recovery is actually able to restore this colonization resistance that's characteristic of healthy subjects. And so this is interesting because it already suggests that colonization is not something that's kind of readily accessible immediately after fermentation. All right, so I mentioned that this response describes 3/4 of the antibiotic taking subjects. So what happened to the rest of them? Well, the remaining 1/4 of antibiotic taking subjects in our study, so this is 5 subjects, they experience what we call lasting responses. And I think you can see from this plot of composition why we call it that. And these subjects, the microbiome shifted dramatically after antibiotics, and then it just kind of stayed at its post-antibiotic state. We didn't see a return to the pre-antibiotic composition. And so people always ask me at this point, do you know why some people have a transient response, but others have lasting responses? The short answer to that is no, we do not. The slightly longer answer is that we have looked with the various factors that are available to us, and we do not see an association of this antibiotic response with prior antibiotic usage, pre-antibiotic community diversity, or the absolute abundance of the communities. This doesn't necessarily surprise me. Microbiomes are known to be very complex systems. We don't have all that many subjects, but at least we're not able to readily identify factors that distinguish them.
It's quite interesting that maybe it's just the colors, but they're like after the treatment in the lasting response, it looks kind of like the pre-treatment in the transient response. Is that just a coincidence?
Yeah, so this is actually a very interesting shift. This subject has a Prevotella-dominated microbiome before antibiotics and a Bacteroides-dominated microbiome afterwards. So microbiome aficionados will be kind of familiar with that particular shift because Prevotella are part of a group of species that tend to be more common among people who live in less industrialized populations and have kind of higher fiber, lower fat diets. And so this shift happening within the subject actually in some ways recapitulates the shift that we tend to see within people as they move to kind of more industrialized areas and adopt these high fat, low fiber diets. And as with that global shift, we don't tend to see the shift back. All right. As I mentioned, we see these different categories of responses, and we can then break down what's happening within these subjects with lasting responses. And so as you might kind of expect, subjects with lasting responses have much more extensive species losses than subjects with transient responses. And so whereas about 30% of disrupted species were lost in transient responders, lasting responders actually had much more of their disrupted species were lost. Actually, something I forgot to mention on the last slide is that the only characteristic that we could find of these lasting responders that was different is that they had bigger responses to begin with. And so bigger response seems to correlate with kind of a longer lasting response. This kind of generally suggests that there's maybe a tipping point phenomenon for the gut microbiome. I don't really count that as an explanation of why though, because we've basically just kind of gone one further link down the explanatory chain. But overall, we do see more extensive disruption, and these seem to lead to long-term species losses. Okay, and so what you might be wondering then is, after these very kind of extensive species losses, do we see additional colonization? Is it the case that kind of new microbes that are coming in are driving this major community shift away from this pre-antibiotic composition? And so we looked for new colonizers within these subjects, and we were able to identify the colonization of new species, which is kind of what you're seeing here, as well as the colonization of new strains of pre-existing species. And so here, as in the example that I mentioned in the primer, we have a stable species abundance, but then we see a shift and the detection of a new strain after antibiotics. And so that's some very nice work by Sophie Walton, who's a grad student in the Good and the Petrov labs. And so overall, we do see this increase in colonization in these subjects that we don't see in subjects with transient response. So what are the new species that are colonizing? Well, for lack of a better word, as we started looking at these new colonizers, we realized that they seemed a little bit weird. They were not the species that you typically expect to be kind of residents of the core gut microbiome. And to kind of quantify this general impression, we looked for the prevalence of different species within the Human Microbiome Project, that's referenced cohort of healthy adult humans. So resident species that were present before antibiotics tended to have high prevalence. That makes sense. Most of the species that you have are species that most people have of the gut microbiome. But when we looked at these early colonizers immediately after antibiotics, we saw that these species had much lower prevalence overall, indicating that they were not kind of commonly found within the gut microbiomes of healthy adults. When we did a taxonomic enrichment analysis, we found that these early colonizers were enriched for different Clostridiaceae as well as Campylobacteriaceae. There were a lot of Campylobacteriaceae. This is a good time just to remind you or reassure you that none of the subjects in our study reported any sort of gastrointestinal illness or side effects from taking this antibiotic. And so all of these changes in gut microbiome composition are occurring unknown to these subjects. So overall, some of these early colonizers, they look like kind of species that are often found in the oral microbiome. Some of them are also potential pathogens, although again, they don't appear to be causing any sort of illness. Overall, our interpretation of these is that although we're just detecting these species shortly after antibiotics, we think that they may actually have been around just below the limit of detection before antibiotics. And we're probably just seeing a bloom of them after the antibiotic perturbation. But that's, again, speculation, because definitionally that kind of suggests that things are below the limit of detection. And overall, another kind of weird feature of this colonization immediately after antibiotics is that it was actually not driving the major community changes that we saw. And so here I'm coloring each species based on whether the strain of that species was something that we previously identified before antibiotics, or whether that event represented strain colonization, either from the cohabiting partner or another source. And so you can see that almost this entire plot looks gray. And so that indicates that actually the massive changes that we're seeing are just the expansion of resident strains, not the influx of new microbes. You might be wondering if there's actually any new colonization at all. I assure you that there is, because we care about this a lot. And so, if you zoom in, you can see those examples of new colonizers. But this really does emphasize that the colonization, or the detection of new strains and species immediately after antibiotics, is something that actually has a very limited effect on the gut microbiome compared to that massive scale of species losses.
Sorry, can I just ask one question quickly?
Mm-hmm.
What was the difference in the total microbial biomass before and after taking that? Because for a lot of low biomass studies, we see a lot of contamination, and also a lot of contamination coming from the oral, like lab environmental contamination during the preparation of the, so for example, if you look into the lung microbiome or any other low biomass habitat, then we do see all this oral bags coming from the person who's preparing the samples and things like that. So did you include negative controls, or how are we sure that this is not contamination and a real colonization effect?
Yeah, we do have negative controls. We didn't see evidence of contamination. We do actually believe that those oral bacteria are real and may reflect transient low abundance. I would argue that they're not contamination. They actually probably are from that subject. That's what we kind of assume, right? There is literally a direct pipeline from the oral gut microbiome. From the oral microbiome to the gut. And it has been shown that in people who go on antibiotics, you can see this kind of transient decline in microbial abundance that's accompanied by kind of the detection of more oral species. We think that this is probably happening to some extent, but as I kind of alluded to extremely briefly, actually these subjects tend to recover the total biomass or absolute abundance of the gut microbiome very quickly, at least as well as we can tell using CFU plating. So that's a very major caveat, but this is not a case where we're totally knocking down the gut microbiome by like several orders of magnitude or something. They are actually relatively normal looking. And we can isolate strains, you know, that works quite fine. So, but yeah, that is kind of an additional complicated factor to keep in mind. All right, so overall, new colonization is not driving these large microbiome shifts that we're seeing. Instead, it's just the expansion of existing resident strains. And you can see that again, if I just sum up the total proportion of the microbiome that's comprised of new colonizers over time, you can see that this is still pretty low for the weeks after antibiotics, although there is an increase compared to control subjects and transient responders. What's really surprising, though, is that when we look out over the period of follow-up sampling, we actually do see a huge increase in the amount of colonization in most of these subjects, barring one strange individual who I don't have time to talk about today, but has an entire figure in the paper. And so within these groups of these 5 subjects who had lasting responses, subjects could actually experience this kind of extensive colonization and kind of large-scale turnover of the gut microbiome. And so we could see new colonizers making up, say, a quarter, up to even 3/4 of gut microbiome composition. The catch, though, was that this colonization took a really long time. Despite these major microbiome perturbations, these new colonizers weren't detected until months and sometimes more than a year after the antibiotic perturbation. And so altogether, to sum up that part of the talk, we see that in these subjects with lasting antibiotic responses, we see extensive strain losses and kind of transient expansions of unusual species, which again may be due to kind of transient low biomass, as well as the colonization of new commensals, but typically only after very long delays. And so altogether, I think that this evidence suggests that even when niches are available after a major disruption, there are still more ecological barriers that limit the pace of colonization. All right, so next I want to address this question of how repeatable colonization is within this household setting. And so here we're really taking advantage of the fact that we're doing this study in human households. And so again, as I mentioned, people who live together sometimes share genetically near identical microbial strains. Using the strain result sequencing methods I mentioned to you, we did detect these shared strains at the beginning of the study. And so some of those households in our study had no strain sharing whatsoever, but then some other pairs actually shared up to half of their gut microbiomes. And so we see this kind of starting amount of strain sharing at the beginning of the study as kind of a baseline level, indicating that there was some prior transmission. And so what's really interesting then is that we can look at the repeatability of these colonization events that we know must have happened sometime in the future. And so what I mean by that is that we want to understand whether strains tend to recolonize the gut after antibiotics. So for example, if 2 people share a strain, one person takes an antibiotics that knocks out the shared strain, then what we want to understand is whether that strain tends to recolonize more quickly from the co-housing partner and drive faster species recovery. And so you might expect this to happen just because, you know, this is genetically near identical to something that was already present in the gut and it's just present in the household. And so what we found was that strains that were not shared at the beginning of the study had a 63% chance of recovery if they were disrupted by antibiotics. And then surprisingly, strains that were shared had a 67% chance of recovery. And this was not statistically significant with our sample size. And so this was really interesting to us. It indicates that after an antibiotic, you can just long-term, seemingly just straight up lose a strain that was previously shared. It's lost, it doesn't come back, even though it's still present within the same household and maintained over time. And so this is really interesting. And again, I think it's evidence that there are these continued ecological barriers to colonization even after antibiotics. And so you might wonder, well, you just spent a while telling me that colonization is very limited and very slow after antibiotics. So what about people, is that just the case that there's not much colonization period? Well, we can actually go a little bit further by looking at the people who had these lasting responses, who did experience additional colonization. And these subjects, they each shared strains with their cohabiting partners before antibiotics. And then we observed in each subject cases in which these previously shared strains were lost. At the same time, over these 1 to 2 years of follow-up sampling, we also observed new transmission events from the cohabiting partner. And so this meant that the strain was not previously shared before antibiotics, it was a new transmission event. And so this suggests that there is some sort of ongoing dispersal between these subjects that actually drives the acquisition of new strains, but seemingly not the recovery of the strains that were previously present. Question. Okay. All right. And so overall, to kind of summarize these results about repeatability, we see that shared strains are lost from the gut microbiome, and there's this lack of recolonization of previously shared strains, even as new strains transmit. And so we think that suggests that there is a strong role for historical contingency in gut microbiome composition. And so overall, these various lines of evidence suggest very consistently, and to me very strongly, that there remain substantial ecological barriers to colonization, even after these really major microbiome disruptions that open up niches within established communities. All right, so for the remainder of our time together, I'm going to talk, first, to return to this original question of whether niche availability is sufficient for colonization in human settings. Our evidence really points very definitively, as I mentioned, towards no. These other factors, dispersal, environmental selection, priority effects, they do seem to have major effects. It's not just that they're operating kind of around the margins to shape colonization. They actually seem to be some combination of these seems to really slow colonization, even after perturbations. All right, so now for the rest of the talk, I will talk a bit about why that might be the case. Why do we see that colonization shows really long delays after this antibiotic perturbation? And to answer that question, I think it helps to return to this conceptual framework that we set up at the beginning. Okay, so there are a couple different hypotheses that we could come up with about why colonization takes such a long time. And one of the explanations that you might come up with is that there might still be low niche availability even after an antibiotic perturbation and large-scale species losses because of functional redundancy. And so in this case, we might expect that resident microbes might fill the niches that were cleared by antibiotics so that there's actually effectively no room for new species to colonize. And so, there is certainly known to be a lot of functional redundancy. Many species can use the same resources. We tend to think that this probably doesn't explain all of the data that we're seeing. Some of the subjects who have lasting antibiotic responses just have this kind of odd oligocolonization. They have very low microbiome diversity compared to typical healthy gut microbiomes. If, the subset of species could typically fulfill all of the roles of the microbiome, we would expect to see lower diversity within non-perturbed individuals, but that's not what we see. The other thing is that we do actually observe colonization, right? So it's not that these subjects are just totally fine with fewer strains strains and species in the gut microbiome. We do actually see that there seem to be a lot of opportunities for colonization, actually, especially with these subjects who go to a lower diversity state. All right, so another possible explanation is that these delays that we observe just represent the time it takes for a new colonizer to reach the limit of detection. And so perhaps microbes actually enter very quickly after antibiotics, but then they're just kind of growing below the limit of detection for a long time. And this is important to think about. There are a lot of microbes in the gut, and we are only able to detect populations that are already quite large. And so instead of this kind of cartoon representation of colonization that I've been showing you, if we kind of make realistic assumptions about limit of detection and the fact that microbes don't have infinite growth rates, then we might have colonization trajectories that look something more like this, where microbes are growing for a long time under the limit of detection, and we see these these apparent delays in colonization just because of that slow rate of growth. And so we think this definitely has some effect in our data, but we argue that it doesn't explain all of the delays that we're observing. And the reason most concisely is because although colonization is delayed after antibiotics, it is also quite abrupt. New colonizers increase in abundance extremely quickly when we do finally see them. And so here's an example of what one of those colonization trajectory looks like. So this person had a strain of Bacteroides johnsonii before antibiotics. It went away, was not detected. And then a new strain was detected, but not until almost 500 days after this antibiotic perturbation. And what was interesting was that it went from being completely undetectable to being at what really seemed to be its kind of carrying capacity within the course of 2 time points, which was in this case about a month. And so what we want to know is how much time was this guy spending below the limit of detection. And so we can just kind of impose some simple assumptions about constant growth rates. And we developed just a simple mathematical model to say, okay, if this guy was growing at a constant growth rate the whole time, then how long did it take for it to grow from this kind of single cell level at the bottom here up to reach the limit of detection? And so, we had a mathematical model, but it essentially does the same thing as eyeballing these trajectories. In short, we can see that this species does spend a long time growing below the limit of detection. But if you assume a constant growth rate, which is a big assumption, that cannot explain the full colonization delays that we are observing. There's still, in this case, something like 200 days that we're not accounting for. And that suggests that there is still some waiting time for the microbe to enter the gut, or perhaps the growth rate is lower at lower abundances. And so I'm showing you just one example here for the sake of time, but actually most of the new colonizers that we saw showed these really abrupt dynamics of colonization, where they increased from undetectable levels to carrying capacity in just a single time point. And so we think that this argues also against the kind of previous explanation that there's just very low niche availability here. The rapid growth does indicate that there's probably a lot of niche availability, but there's still some other sources of delays. All right. And so another hypothesis to consider is the extent to which dispersal limitation and environmental selection limit colonization. And so another way of framing this just more simply is, are most microbes we encounter already dead? I think very few people would dispute that we encounter microbes all the time, but it might be that through the filter of environmental selection, the number of live microbes that reach the gut is actually quite low. There's reasons to think this. When we look at probiotic strains, it's estimated that 20 to 40% of them actually reach the gut alive. And these are strains that we are trying pretty hard to keep alive. In contrast, your average microbe that's been hanging out on a surface for a while, it may have been long dead by the time it is consumed by you. And so it would have a very low chance, well, 0 chance in that case, of colonizing in the gut. And so this is something that we think is quite important. It's incredibly difficult to directly measure. So we think that this has some amount of effect on our data. There are a couple observations that I want to highlight that do kind of complicate this picture, though, of dispersal limitation. One of that is, I already told you, previous shared strains that were previously shared between cohabiting partners failed to recolonize, even as new strains transmitted between these subjects. And so that new transmission does show that there's some ongoing amount of microbial exchange, but just still not the not enough to promote the colonization of these previously shared strains. And so there is at least some dispersal acting in these subjects. Another kind of interesting observation that I want to point out, and I think that this is at least one of the 2 most interesting things that we saw in the study, is that multiple commensal strains sometimes undergo really what appear to be coordinated expansions months after antibiotics. So under very simple models in which you're just kind of waiting for live microbes to enter, but they grow really rapidly once they enter the gut, you would not expect to see multiple strains colonizing at the same time. But that is, in fact, what we see. And so in some of the subjects with lasting antibiotic responses, we detected a large proportion of colonizers at the same time. And as you can see from these colonization trajectories, they really seem to have very, very similar behaviors. And so again, this at least complicates our picture of dispersal limitation. This may suggest that new microbes disperse collectively, and so you may just kind of get a large propagule that contains a lot of different species. That's a pretty reasonable assumption, I think, in the gut. There's still this open question of why species that arrive together would grow at similar rates. That's not entirely obvious. And so another possibility, which is non-exclusive with collective dispersal, is that the microbes may facilitate each other's growth. And so you may, facilitative interactions may drive these coordinated expansions. All right, so yes.
So for this plot, have you looked at the absolute abundance of this?
We have not. That would
Because,
be,
the relative abundance of this correlated behavior might be that the other bugs abundance change.
yes, that is possible. I think probably, yeah, this kind of continued change could be due to kind of other bugs, but I do think kind of this coordinated increase from limit of detection to near carrying capacity is kind of surprising. All right. So at this point, you might say, what do you actually think is going on? I presented a couple explanations that I think are not complete explanations of our data. And so based on the full sum of all of this data, we would argue that there appear to be strong priority effects in the gut microbiome. And so the idea, one of the kind of pieces of evidence that I think points to this is the fact that resident strains recover very rapidly in the week after antibiotics, but new strains take weeks or months to colonize. And so there's this discrepancy in time scale between the recovery of something that was already there and the colonization of something that's new. And so another way to think about this is that for resident strains, what we presume is happening is that antibiotics are knocking them down below the limit of detection, but they're able to recover from kind of sub-limit of detection abundances after the antibiotic is gone. In contrast, these new strains are actually starting from zero abundance in the gut microbiome. And so, they take weeks or months to colonize. The difference between these two scenarios is what happens at these really low microbial abundances. And so I've already told you one of the major factors in play, which is that microbes first have to disperse from one host to another. That we do think is a key barrier. But I think something else that's really important to consider is the possibility that these initial stages of establishment are really important in limiting colonization. And so priority effects give kind of an incumbent advantage to existing residents over new arrivals. And so they would tend to especially slow these stages of initial establishment. Using our data alone, we cannot kind of conclusively demonstrate any mechanisms of priority effects. But there are possible mechanisms in the literature that could give rise to these phenomena. And so there could be inhibitory priority effects that arise through bacterial antagonism. So this is where resident strains and new strains may have kind of incompatible toxin-antitoxin systems that actually prevent colonization. And we see some circumstantial evidence that this may be going on in some of our data. Additionally, there could be facilitated priority effects. And so biofilm formation involving facilitative interactions between members of the same species could also contribute to priority effects. So could cooperative or cross-feeding interactions between members of different species. And again, this may be one explanation for why we see multiple microbes seemingly reaching high relative abundances at the same time. All right, and so I think that our data suggests that these mechanisms, potential mechanisms of priority effect, are very well worth additional study. All right, so just to sum up everything I've discussed, I want to leave you with a parting thought about why it matters whether we actually distinguish between ecological forces. You might say, yeah, this is all kind of ecological mumbo jumbo. Anyway, what does it actually matter whether one thing or another actually shapes the gut microbiome? Well, I think priority effects in particular are a mechanism that has very strong implications for how we think about the microbiome, because priority effects in particular would create very strong contingency in microbiome composition. And again, I think this has implications for how we think about the kind of change and development of the gut microbiome over the course of life. And so if we're in that niche availability regime where selection is strong, dispersal is fast, and there are limited priority effects, then what that means is that everyone's gut microbiome is kind of in the survival of the fittest race. and you, each of us has kind of the best of all possible gut microbiomes because we are constantly sampling many of the microbes around us that have kind of fair chances to compete against residents and stick around in the community. And so this kind of strong selection regime means that we would tend to have microbiomes that are very, very well suited to the host as well as to kind of diet and other environmental influences. In contrast, if priority effects are in fact operating strongly in the gut microbiome, then that takes us into a very different regime. That indicates that there's a lot of hysteresis or state dependence in microbiome composition. In other words, instead of microbes being in your gut because they are the best of all possible microbes and kind of competed successfully to make their way in, instead the kind of concise explanation of this is that the microbes in your gut are there because they're there. Like they were already there, they got there at some point, perhaps during a critical window, like antibiotics that opened up new niches. But after they're there, they just kind of continue to stay there because there's just very high barriers to entry for anything new. And so this kind of paints a very different picture of why we have the gut microbiomes that we do. It suggests that selection may actually have selection may be less efficient at operating in the gut microbiome, and the microbes that colonize in certain circumstances may not manage to colonize again. And so I think that distinguishing between these different scenarios does have profound implications for how we think about the gut microbiome in general, and in particular, how we think about designing therapeutics that are actually able to successfully perturb gut microbiome composition.